We're going to move on now to work package number 3, which is entitled How the Location of the Microbiome and its Interaction with Colorectal Cancer Cells May Impact Potential Therapies. And for this session, we are going to welcome Marielle Giannakis and Marielle Santos McLeod. Professor Giannakis is a physician scientist at the Dana-Farber Cancer Institute and assistant professor of medicine at Harvard Medical School. Marielle also has an active clinic caring for patients with gastrointestinal cancers. Marielle was diagnosed with young onset stage 3 colon cancer in 2017. At the time of her diagnosis, she was shocked as to how little information was available regarding colorectal cancer in young adults, and she has set out to change this. Today, she is the family support navigator at Colorectal Cancer Alliance. Together, Marielle and Marielle will explore the relationship between colorectal cancer microbes and specific cell types of the colon, and how this is leading to new potential therapies.
So welcome to Work Package 3. We're going to talk about mapping the cellular distribution of colorectal cancer microbiome. And we're here today with Dr. Marielle Giannakis and Marielle McLeod, our wonderful patient advocate. And we're going to talk about the hypothesis first of this project, and that is that bacteria from the colorectal cancer microbiome are actually directly associated with colorectal cancer cells. So Dr. Giannakis, can you explain to us a little bit about the goals of this project and how that hypothesis plays into your research?
Absolutely. And first of all, thank you so much for having me. And it is my pleasure to talk to you about the work we do collectively in Work Package 3. And I'm representing, of course, additional investigators that could not be present at this call, including Dr. Matthew Meyerson, Dr. Paul Nucifor, and others, as well as our amazing patient advocate, Marielle, who's joining us today. So yeah, the interesting, our hypothesis is that there is definitely changes among individuals pertaining to the location of the cancer-associated microbiome. We don't know exactly where microbes reside in this vicinity, in this neighborhood of cancer cells and other cells that surround cancer and normal colon tissues. When we think about the geography of the colorectal cancer microbiome, we can think about it in many levels. One is that, of course, patients and individuals from different countries can have a vastly different collection of microbes depending on things like their diet, the location they're residing, and so forth. But then one can zoom in a little bit and look at the geography of the colon itself. And there's definitely differences between microbes, between the different sides of the colon, right side versus left side. In this work package, we are taking an even deeper look and trying to define the relationship between colorectal cancer microbes and specific cell types. Are they joined? Are they adherent to? Are they inside cancer cells? Are they next to or impacting immune cells of the colorectal cancer? By defining the location of these microbes and how they impact cancer cells and other surrounding cells, we definitely hope to impact patients by devising new ways to intervene with the microbiome and improve outcomes for patients with colorectal cancer.
When we talk about the location of bacteria within the colon, are we talking about any bacteria in particular? Are there many different types of bacteria? What type of bacteria or microbes are we finding with inside the bowel in the microbiome?
Yeah, that's a great question. First of all, we should say that the colon, even without cancer, normal human colon is the site of highest density of bacteria in the world. So you will never find more bacteria per size than you will find in the human colon. But beyond that, we know there are some microbes that are enriched. They are more prevalent in colon cancer than in normal colon. Sometimes we refer to these bacteria as oncomicrobes because they are, again, found more frequently in cancer tissues than in normal tissues. There are some species, some individual bacteria that are more prevalent in cancer, such as Fusobacterium nucleatum. That's a bacterium that has been described by our groups and others to be enriched in cancer, as well as a few others, like Bacteroides fragilis and so forth. The challenge is to figure out if they are innocent bystanders or they're actually involved in either causing cancer or more rapid development of cancer or even cancer metastasis.
And are they consistent? Are these bacteria consistent throughout the colon? Do they vary in intensity, for example, on one side versus the other side? And, do they travel with cancer cells when they metastasize? What's the story of the Fusobacterium?
Yeah. And of course, those are some of the exact questions we want to answer, right? And we're still in the process of understanding that. I will say that there's a lot of things we need to learn about the microbiome and its relationship with colon cancer. But we do know that, yes, there are some bacterial species like Fusobacterium that I mentioned that are more prevalent in one side of the colon versus the other. So these specific ones are typically found on the right side, and they're also associated with specific molecular subtypes of colon cancer. Why, we don't completely understand. Whether they travel with metastasis or not is a great question as well. There were some evidence that paradoxically, colon bacteria can be found in distant metastatic sites traveling together with cancer cells, but these results were preliminary and we still are trying to figure out the role of the microbiome, not only in cancer development, but also cancer metastasis. This is why defining where these microbes are in relation to cancer cells, but also other cell types of the environment is important. It can lead to insights that then will allow us to devise ways to interfere, perhaps intervene, right, and prevent colorectal cancer development, progression, or even metastasis.
And how are you actually doing this? What is the methodology in order to track these bacteria and find them and geolocate them, so-called, within the colon?
Yeah. So we are fortunate to work with a large team of specialists from not only medical oncology and basic science, but also pathology and so forth. There's several ways. One is through examining the tissue from cancer, as well as normal as a control, and looking with techniques that visualize these bacteria in the tissue itself. And some of those are things like, they have fancy names like hybridization techniques or immunofluorescent techniques, but the basic idea is that you have probes or tags that will locate and highlight these bacteria in the cancer tissue. There's also some newer technologies that we've been working with, such as characterizing the environment of the cancer at individual cell resolution. So we're breaking up apart these cancers into their individual cell types. And then we're asking whether imprints, DNA fragments from bacteria are found within those cell types. So there's a few technologies that we use to really pinpoint the exact location of this bacteria and how they may affect these other cell types, whether they are cancer cells, immune cells, stromal cells, or vascular cells.
So a very important part of your team that you mentioned at the outset, which helps to shape your research, is both as a researcher and as a clinician, having the patient perspective. And Marielle is with us today. Marielle, why is it important for patients and patient advocates to be engaged in research, and particularly in this particular project?
In a previous conversation that you and I had, Barry, I mentioned that knowledge is power. And honestly, as a patient advocate, there's nothing better that we can bring to the table than outside knowledge that we have gained by participating in research packages not just for the grand challenges in OPTIMISTICC, but also the one that Marios is working on in regards to the microbiome and how definitely can impact further research on, being able to pinpoint the specific cause and development of colorectal cancer. However, as a patient advocate, what I have loved about being a part of this work package is that I get a front row seat and kind of like a sneak peek into the development of any potential heighten and increase in treatment that our patients might be able to benefit from. And I actually get to bring that back to my team and, selfishly provide the information that I have learned from Marios and be able to share that with him and how we can further impact our patients.
And you have a personal interest in this as well. Maybe you could tell us a little bit about that.
I just wanted, one of the questions that I do have is in regards to being born in a different country and moving in specifically into the United States where young onset cancer cases are sadly on the rise, especially those at 2% in the last 2% a year, especially within the last decade, is do you see any possible impact outside environmental being mostly part of it as one of the determining factors in your research?
Yeah, no, that is a fantastic question and a point. And you also bring up this epidemic really of young cases of colorectal cancer, patients diagnosed less than 50, and unfortunately in many cases, much younger than that. And I will say we don't completely understand the reasons behind this, but what you're alluding to may be actually an important component, just because we know this phenomenon of young onset colorectal cancer is relatively recent. And we know human DNA doesn't really change as fast as other factors, including the microbiome, that are impacted by the environment. So just for that reason alone, for the fact that the microbiome can change both by moving between countries, by eating different things, and so forth in ways we don't understand, but this makes it a prime candidate to examine for its role, perhaps, in young onset colorectal cancer carcinogenesis. And we do have efforts as part of this work package, but also as part of some other work packages in the OPTIMISTICC team to really specifically define the microbiome of young onset colorectal cancer patients and its interactions with dietary, macro-environmental and micro-environmental factors.
And when you're mapping out the atlas, so to speak, of the microbiome within the bowel, do some of these factors, whether it's diet or physical environment, impact the mapping of the microbiome?
Yeah, that's a great question, and unfortunately, I don't think we have the answer to that yet, because we're still trying to navigate our way around and map, as you said, the location of the microbes in relation to other cells, just at baseline in several cancers. But absolutely, the next step would be to see how certain features impact that distribution. And those can be factors of the cancer cells themselves, including different mutations that are found in cancer cells, different types of colorectal cancer, as well as exogenous factors like diet, for example. Yeah, we don't know that yet, but that's definitely something we aspire to understand.
I know that as a clinician and dealing with patients on a daily basis, patients are really first for you. How has having had a patient advocate in this research project impacted you and of course your team, your entire lab together?
No, it has been immensely enriching to our research efforts to have Marielle on. Our motivation and mine personally certainly comes from trying to do better for our patients. But it's not only that, it's also that, again, her questions, her approach help refocus us to really the main goal of this project, which is to try to figure out ways to either prevent colorectal cancer, slow down its development, and come up with better therapies. I also believe Marielle's specific questions have been very insightful and serve really to illustrate the need to clarify our hypotheses, focus our research to get to what really matters.
So thank you both for participating today. And I look forward to working together with both of you over the course of this project.
Focus on what really matters. That was a wonderful final statement from Marielle at the end, which is really the true value in having patients and researchers collaborate on the excellent work that's being done. Thank you to Marius and Marielle for most particularly highlighting the epidemic was the word that was used of colorectal cancer among young adults, and some of the investigations that are being generated around factors which are contributing to that.